Hello everyone and welcome. In this video we're checking out variant 1 of May-June 2021 paper 2. This is the first paper 2 we're checking out together. So let's go ahead and get started with question number 1. Also, make sure to check out my free notes I have available for you in the comments down below for chapter 2 AS Bio and chapter 1 A-Level if you are an A2 student. Come on, question number 1. Figure 1.1 is a transmission electron micrograph of cells from duckweeds and whatever the name is. Okay, we have calculate the actual width of the cell labeled X. Write down the formula you use to make your calculation. Show your form working and give your answer in micrometers to 1 decimal place. Okay, fantastic. So firstly, the formula, we have to refer to the famous triangle. Okay, I'm not sure I could write here with my finger, can I? No. Okay, so let's go ahead and use the keyboard. Okay, so remember the magic triangle is going to contain magnification, image length, and actual length, where magnification will be on the bottom and the image length will be divided by actual length. So in the case where we want actual length, please make sure to draw out your triangles. I can't really do it right now. However, I will write down the formula for you right here. So it will be actual length equals image length divided by magnification. Now in this case they're actually asking for what's it called, actual width. So let's change these and swap them out for width. Okay, now we're done with the first part of the question. I'm going to go ahead and do the calculations. Now, to do the calculations here, you have to give your answer in micrometers. What we have, however, is an image where when we measure, we're going to have to use centimeters. You're going to write down the measurement in millimeters, but you're going to measure it in centimeters. So for example here, remember my ruler is really not accurate because the page could vary, but for you the past papers are very close to how the exam would come. So you would have to use your past papers in order to figure this out using your ruler. I would get 1.8 centimeters, which is 18 millimeters. Remember to multiply by 10 in order to convert from millimeters to centimeters. It's really not that complicated. Now what I'm going to do is I'm just going to go here and I'm going to show you guys how it's done. So basically I have 18 millimeters, so let's write this down. Image width equals 18 millimeters, and what's it called, I already have the magnification given to me at the bottom of the picture, so let's go ahead and look at that. So you have 4,275. So what you want to do to calculate actual width is to divide 18 by 4,875. No, actually, that doesn't make sense really, and the answer should be in micrometers. So how do you go from millimeters to micrometers? Well, you need to convert the image width from millimeters to micrometers. And remember that 1 millimeter is actually equal to 1,000 micrometers. So we need to multiply by 1,000. So we have here 18,000 micrometers. Okay, you guys get the point? Because this is a small thing, you need to write down the units next to your final answer. Okay, so it will be 18,000, and make sure to write this down before just jumping into writing it in formula. 18,000 divided by 4,875, let's see what we get. 18,000 divided by 4875, if I'm not mistaken, it should be approximately, oh actually it's 4,275. Okay, so 4,275, you got 4.2 micrometers. So actually I get 4.21 micrometers. So 3 significant figures is what you would want to aim for. And since he already gave you the answer, you don't have to write them down yourself. Letter B, however, talks about table 1.1, which lists some biological molecules found in plant cells. Complete table 1.1 by choosing one letter from figure 1.1 that indicates a cell structure where biological molecules are found. Okay, so this is about chapter 2. So if you're looking for classified questions for chapter 2, this is one for you. Biological molecule, DNA. Where is DNA found? It's common knowledge that DNA is found in the nucleus, but it's also found elsewhere. So here we have B, where DNA is found in the nucleolus, and that's true. Could we choose C? C is the chloroplast, or is it? If you zoom in here, or if you just look really tightly upon your past papers, you'll find actually that C points to the thylakoid membrane, or the grana, and granum. And so in this case, obviously granum do not contain what's it called, DNA. However, if we if we were to choose chloroplast as a whole, then sure, chloroplast does contain circular DNA. But in this case, I will just choose B. So let's go ahead and write B right here. Okay. Then he asks about cellulose. Cellulose is present in the cell wall. Let's look, where is the cell wall labeled? It's E. Okay, fantastic. So this would be E. Okay, now he's asking for phospholipid. Now, obviously, if this were any other test, I would have said if, for example, chloroplast was an option, you would write both B and the chloroplast. But since here he says specifically to only choose one letter, then you can't do this. However, in another question exam where he might not say this, you have to write down all the options available, okay? Phospholipid is probably present in membranes, so we could choose, for example, the thylakoid membrane. C, we could choose the cell membrane. However, it's not really pointed. We could also choose the nuclear envelope or the nuclear membrane, A. So it's really up to you which one you choose. I'm going to choose C. Okay. And lastly, he asks about histone proteins. Histone proteins are found in DNA. DNA is found in the nucleolus, and therefore the same option would be for the first and last questions. Okay, state the name of a cell structure visible in figure 1.1 where ATP is synthesized. The name specifically of the cell structure. Now, what we have here, obviously, have a labeled cell structure, a visible cell structure. Okay. A visible cell structure here would be, we have what's it called, chloroplast. I can't really see any mitochondria, so I'm just going to say chloroplast, because actually chloroplast does synthesize ATP during what's it called, photosynthesis. So I would say chloroplast. It will be there and then later on used as well in the same process. So name a cell structure that produces mRNA. mRNA is produced in the nucleus, so we could just go ahead and write this down. Nucleus. Okay. Then he says, describe the evidence from figure 1.1 that shows that the image in the transmission electron micrograph okay, so how do we know that this image taken is a transmission electron micrograph, not a what's it called, one taken by a light microscope? So in this case, you could clearly see chloroplast, and you could clearly see the membranes inside the chloroplast. And for your information, membranes are only 7 nanometers thick, and for that we require really, really high resolution. Now, in questions like this, it's really important to define resolution. It's really important to write down why what's it called, electron micrograph has a higher resolution than the light microscope, and why therefore this is an electron micrograph. So let's go ahead and write all that information down. So let's go ahead and write this. Firstly, resolution is the ability to view 2 points as separate. To resolution, and I would write here define, and obviously you don't have to write define, but like, okay, so is the ability to view 2 separate points or objects as separate, okay? Because if, for example, you have a camera and this camera has such a low resolution, then this means that you might see 2 things that are 2, for example, things, 2 buildings, they merge into one, and the image is really blurry and unclear. However, when the resolution is higher and higher, we're able to see things that are so close together as 2 different points. And so in this case, we're able to do this because the electron microscope has a higher resolution than the light microscope. So let's write this down. Electron microscope has a higher resolution than the light microscope, and you can also write down the numbers if you want, where the electron microscope has a resolution of 0.5 nanometers. So anything that's called further than 0.5 nanometers apart will be seen, like the membranes, which are 7 nanometers apart. 0.5 nanometers. The light microscope, however, has a minimum, we're talking minimum resolution, which obviously maximum in terms of like how great it is. So you could see how like that could get confusing. Light microscope has a resolution of 200 nanometers when using purple light, okay? Now, obviously here you could see things. Now, when do I stop writing, you may ask? This question has 2 marks. I prefer that you write 3 points, if not 4. Okay, so I would say here that the electron microscope has a high resolution. Also that you can see thylakoid membranes of chloroplast, which are obviously, okay, excuse any spelling errors, but you can't do this. You can't do this in your exam. I'm just typing really fast. So membranes of chloroplast and any other membranes, which are, 7 nanometers apart. You can also see the cell membrane, you could also see the nuclear envelope, you could see lots of things. So which are 7 nanometers apart and not visible using the light microscope. Okay, now I'm going to move on to the next question. Okay, question number 2. Figure 1.2.1 shows 3 molecules of water. So you have 3 molecules of water, as you could see, and they're all bonded together by hydrogen bonds. He's asking you to describe the hydrogen bonding that occurs between the water molecules shown in figure 2.1. Of course, okay. Firstly, in such a question, you could just go ahead and start by defining a hydrogen bond. A hydrogen bond is a weak bond, that forms between between 2 water molecules. And we're talking about it's a bonding between the partially negative charge on the oxygen atom with a partially positive charge on the hydrogen atom from another water molecule. So you have to write this down. Okay, come on. Between 2 water molecules, or let's go ahead and say instead between the partially positive charge / you could also always say delta positive, okay? And if you know the delta sign, even better. Okay, delta positive on hydrogen atom of 1 molecule with the... Okay, let's move on to the next line. With the partially negative on the oxygen atom of another, water molecule. So that's the definition of a hydrogen bond. Okay, so that's point 1. You have 3 points to go. Okay, now here is asking you to describe the hydrogen bonding that occurs between water molecules. What else do we know about hydrogen bonds? Why do water molecules form hydrogen bonds? Well, water molecules are polar, okay, and what does it mean to be polar? Water molecules are polar, which means that they have dipoles. Okay, so they have dipoles. What does it mean to have dipoles? Dipoles just means a uneven distribution of charge. So since they have dipoles, so that means it's an uneven distribution of charge, which is why we have a partially positive charge on hydrogen and partially negative charge on oxygen of charge. And that's why hydrogen bonds are going to form. And we can also write down and mention here that each water molecule is able to form. Moving on to the next line. Okay, 4 hydrogen bonds with obviously 4 different molecules. Okay, now let's move on from this question to B. The human enzyme salivary amylase is composed of 1 polypeptide. Figure 2.2 represents the structure of a molecule of salivary amylase. So you could see here, salivary amylase is, you could see that there's a tertiary structure going on. There's also an alpha helix and some beta-pleated sheets. I wish I could point these out for you. This over here under select all is going to be the alpha helix, and then this over here, do you see the blue? Yeah, right here, this is the beta-pleated sheet, which basically is just a bunch of parallel lines, as you can see. Okay, moving on. He says, explain the role of hydrogen bonding in maintaining the secondary structure of proteins, such as the salivary amylase. Okay, well, the role of hydrogen bonds is that hydrogen bonds actually are what maintain the secondary structure. And when talking about secondary structure, we're talking about the alpha helices that we saw, about the beta-pleated sheets that we saw. So just refer to the structure that we had above. What's the secondary structure that we see there? Beta-pleated sheets and alpha helices. And therefore, I would say, oh yes, hydrogen bonds maintain those. Hydrogen bonds maintain the alpha helices and beta-pleated sheets of amylase. Okay, because it's simply a one mark question. It just requires you to refer back to the diagram. Okay, explain the role of hydrogen bonding in maintaining the tertiary structure of proteins such as salivary amylase. Sure. So firstly, we need to understand what is tertiary structure. So obviously we need to define tertiary structure. Tertiary structure is supposed to be here we go. Okay, so yes, it's the folding and the what's it called extensive coiling of the polypeptide chain upon itself, forming this 3D structure. Right, so what's it called? How is the 3D structure maintained? Maintained by the hydrogen bonds, maintained by ionic bonds, disulfide bonds, and what's it called? Hydrophobic interactions. So now when we're talking about hydrogen bonds, how do hydrogen bonds form? Hydrogen bonds are going to form between the what's it called carboxyl groups and amine groups, which are like, these are not really specifically the carboxyl group amine groups that are part of the amino acid. Rather, they are when what's it called an amino acid has an R group of that sort. Then these bonds are going to form between what's it called the oxygen and the hydrogen of each of the groups. So let's write this down. Okay. And this is a weird box. Let's just make it bigger. Okay. So I can't type, can I? So let's write all this down. Firstly, what's a tertiary structure? Wow, okay, tertiary structure is what's it called? The extensive coiling and folding. Okay, it's really weird how the size grew, but anyway, what's it called? Folding of a polypeptide chain upon itself, forming a 3D structure, which is maintained by what's it called? Things like hydrogen bonds, where which is going to happen, which happens between, here we write amino acids with carboxyl, not carbonyl, carboxyl, and amine R groups, where the the oxygen, which is partially negative, is attracted to the hydrogen. Obviously here you could write down the oxygen of the carboxyl group, attracted to the hydrogen of the obviously here hydrogen, which is partially positive, of the amine group, and that would be the hydrogen bond. Outline the importance of water as a solvent in plants. Okay, so water is really important as a solvent because it's useful in transport, for example, in translocation and also in transpiration. It's really useful also as like a solvent for chemical reactions, a place for chemical reactions. Off the top of my head, I can also think of like water being useful in plants for photosynthesis, right? Water is going to be used in photosynthesis and what's it called, like as a reactant actually, but it's also going to be present like in what's it called it's going to be the place or the medium for the reaction in general. So yes, it's really useful as a solvent. So let's go ahead and write these things down. So obviously water is a polar molecule. Don't forget the properties of water. It's a polar molecule, so it's able to dissolve. Likewise, polar molecules able to dissolve polar molecules, so it's useful in transport, such as in for example in translocation. We're talking mass flow here. Okay, in translocation in the phloem, mass flow would require water. You could also mention here in the xylem, okay, water will dissolve mineral ions, okay, and transport them. In the phloem, you could mention, for example, that water would dissolve amino acids. Now here, for example, what happened? Okay. So this just moved, sorry about that. Okay. You could also mention that water is a solvent or medium for chemical reactions, generally all chemical reactions, and it's also a reactant in photosynthesis. Photosynthesis. So just think of properties of water, write all that you know related to plants down, okay? Moving on. Whisking tubing can be used to investigate the properties of cell membranes. A student carried out an experiment to investigate osmosis using whisking tubing. An outline of the investigation is shown over here so you could see what's going on. You have whisking tubing and you can see the meniscus shows that the water level is certain here, and then what's it called, once you put it inside this tube after 20 minutes or certain time, I don't know how much, basically you're going to look at the level and you can see how this is going to change. So let's see, 6 pieces of whisking tubing were filled with 10 centimeters cubed of different concentrations of sucrose solution, 0.0, 0.4, 0.8, 1.2, 1.6, and 2.0 mole per decimeter cube. The height of the meniscus of each solution in whisking tubing was measured, and pieces of whisking tubing were placed into test tubes for containing 15 centimeters cubed of 0.9 mole per decimeter cubed sucrose solution. After 20 minutes, the pieces of whisking tubing were removed from the test tubes, and the height of the meniscus in each was measured. So the height of the meniscus after time is going to be measured, and you can see here the results shown. So we're looking at the difference in height and the concentrations how they vary. So when you change the concentrations, the difference in height will change actually from negative to positive, with a switch happening between 0.8 and 1.2 mole per decimeter cubed. And that makes sense because at 0.9 mole per decimeter cubed, the concentration inside the whisking tube will equal to the concentration of the sucrose solution. So at that point we'd have zero change in the height. But before that, osmosis is going to happen because there is a difference in the concentration gradient. There will be a difference in the water potential. So osmosis will happen. So let's see what does letter A ask us? Okay, the whisking tubing used by students was not permeable to sucrose. Could you explain the results in table 3.1? Okay. Whisking tubing was not permeable to sucrose. How do you explain the results? Okay, so here in order for us to explain it, we need to just describe osmosis, how it happens. We need to explain why why the water movement moves in the way it does according to osmosis. So let's see, according to values as well. We could just obviously use values in this case because I think it's it's actually just a 3-mark question. Okay, so yeah, I think we could just start by saying that the osmosis is basically the movement of water from a region of higher water potential to lower water potential down water potential gradient through a partially permeable membrane, which is the whisking tubing. And so in this case, water is obviously before anything below 0.9, we're talking 0.8, 0.4, 0.0 mole per decimeter cube. These are going to be the points where the concentration inside is lower than the concentration outside. So the water potential inside the whisking tubing is much higher than the water potential outside the whisking tubing, and so water will leave the whisking tube by osmosis, and so the water level will decrease inside the whisking tube, and that's the explanation. Now when you increase when you increase the concentrations above 0.9, that means 1.2, 1.6, 2.0 mole per decimeter cubed, and at that point you're going to have a positive difference because the water potential inside will be lower than the water potential outside, since the concentration inside is higher than the concentration outside, and therefore water will move out of the solution into the whisking tubing, increasing the water level, and thus a positive change in the water level. Okay, so let's go ahead and write down everything we just said as it is. Okay, so let's see what we can do here. Okay, interesting. How do I fix this? Here you go. Okay, so firstly, as I mentioned, osmosis is the movement of water from a region of high water potential, water from higher water potential to lower water potential down a water potential gradient through a partially permeable membrane, and in this case it's really the whisking tubing. At concentrations inside whisking tubing below 0.9 mole per decimeter cubed. For units, I'm just going to write X, and you guys should understand that this means mole per decimeter cubed because it's really long for me to just write it down every time. Or the concentration of the solution. Water will what's it called? Yeah, the concentration of the solution. And here we mentioned examples that we have here. What's it called? What are they? 0.0 with the units. And then you have 0.0, 0.4, 0.8, 0.0, 0.4, 0.8. Water is going to, water will move from the solution, which has, from the whisking tubing, lower concentration means higher water potential, into the what's it called solution, decreasing the water level. And then opposite will happen. Water will move, water level increases in concentrations above 0.9 x for unit. Okay, you don't write x, I just mean x as this means molar per decimeter cubed, because I can't really write it down here. So it's called above 0.9. Let me try to just, yeah, okay. So above 0.9, and then you mention examples, which are 1.2, as well as 1.6 and 2.0. 1.6, 2.0. What happens here is that water will move. Now, since the level increases, this means that the water will move down a water potential gradient from the solution into the whisking tubing. And that's it. Moving on, we have when red blood cells are placed in water, they are destroyed by bursting. The students are also investigated how red blood cells are affected by immersion in solutions of sodium chloride of different concentrations. Blood samples of the same volume were added to the solutions of sodium chloride in separate test tubes. Interesting. So after 10 minutes, the student took 0.1 centimeters cubed of blood samples from the test tubes and estimated the percentage of red blood cells that had burst in each sample. So you can see here we have concentration of sodium chloride solution against the percentage destroyed, and you could see 100% will be destroyed when the concentration is actually below specifically 0.04. Could you describe and explain the effects of red blood cells on immersion on different concentrations of sodium chloride as shown in figure 3.2? Sure thing. Okay, let's look at figure 3.2. Just analyze this in our heads and then write down any analysis we have there. Now we have to do two parts in this question, not just one. We need to describe and we need to explain. Now to describe it, I would say that here we have 100% bursting, so I would say that anything below this concentration will burst, and then between we have a curved relationship where the percentage keeps decreasing, and there will still be bursting, but between the specific concentration of 0.04 and I think this is 0.14 mole per decimeter cubed, the concentration or percentage of red cells will decrease at burst, and then lastly anything above 0.14 will not burst. Okay, now this is a description, and I think because this is a format question, let's try to dedicate 2 marks to description, 2 marks to explanation, and try to even increase them further. So now, explanation, I would obviously explain the concept of osmosis. I would say osmosis is the movement of water down water potential gradient and define it, sure thing. But I will also have to say why the red blood cells burst. Red blood cells are going to burst because there's so much water moving into the cell that this will increase pressure, or let's say, partial pressure, internal pressure, so much that the red blood cells burst. And obviously cell membranes can't handle this kind of pressure. That's why plant cells do not burst under the same kind of pressure, because they have cell walls, and cell walls are able to maintain pressure, that are able to handle that kind of pressure, and that's why plant cells simply become turgid, they don't burst. Our cells, however, are going to burst. So we need to also mention the point about cell walls. I think it could be relevant in this case. So let's go ahead and describe and explain the effects of red cells on immersion in different concentrations. So let's write down everything we just mentioned, if not even more in more detail. Okay. So explanation, osmosis is movement of water down. So let's define osmosis. Okay, so of water from tier, of course, tier of water from region of higher water potential to lower water potential. Of course, it's potentiometer. You guys, please ignore the potentiometer situation. Okay, to lower water potential down a potential gradient or down a water potential gradient. Okay. Now moving on here, water is going to move. Okay, moves from the solution to the red blood cell, and when the concentration is so low, when the water potential outside is so high, so much water moves in. So much water moves into the cell, which obviously the cell membrane can't handle the pressure, and red blood cells don't have cell walls. Okay, I'm really sorry about all the spelling mistakes. To to maintain or to handle the pressure, and so cells burst. Now that's explanation. I think we're good on explanation. Let's go ahead and describe. Okay, description, I would say that, as we mentioned, below a concentration of 0.014, 100% of red blood cells are going to burst. So below 0.04, concentration, you obviously need to write down the units, mole per decimeter cubed, 100%, or let's just write all red blood cells burst. Okay. And now between 0.04, right, between 0.04 mole per decimeter cubed, okay, and what's exactly, and 0.14 mole per decimeter cubed, okay, so what happens to the the percentage of red blood cells bursting? Decreases proportionally with the decrease in concentration. With the increase. So we could actually say it's inversely proportional, but yeah, with the increase in concentration / you could also say decreases in a curve. Okay. You could mention here an example, a figure, 2 figures actually, because we're describing how it will decrease proportionally. So I would say, for example, and we move on to the units here. For example, what you would write, I will not write this right now because we need to move on, at a concentration of 0.06 mole per decimeter cubed, 80% of red blood cells burst. But then when you increase the concentration to 0.1 mole per decimeter cubed, you're going to find that actually only 28% of red blood cells have burst. And so that's the way of demonstrating that information. Lastly, any concentrations above 0.14 mole per decimeter cubed, red blood cells don't burst. Okay, and that's it for this question. Moving on. Question number 4. The induced fit hypothesis and lock and key hypotheses are used to describe the mode of action of enzymes. Could you explain the induced fit hypothesis? Sure thing, let's explain it. So, the induced fit hypothesis is going to be where the enzyme doesn't exactly have a complementary active site to the substrate. So, what happens is here the enzyme is going to undergo a conformational, or it's change in shape, where it's going to, fit, or let's say mold into the mold around the substrate, and therefore they're going to be able to form the enzyme substrate complex after all, and then the enzyme is going to catalyze the formation of the products, and then once the products are going to leave the active site, the enzyme will return back to the way it was. So this is exactly how the induced fit hypothesis works. So let's go ahead and write this down. Okay, so the enzyme active site not really complementary to the substrate binding site, and so what happens is, as we mentioned, enzyme changes shape or molds or undergoes conformational change in shape, whatever you want, okay, to what's it called, fit substrate, and then enzyme substrate complex is going to forming the enzyme substrate complex. And lastly, you're going to talk about how the enzyme catalyzes the formation of products, which leave the active site. Enzyme returns to the way it was. Okay. Radish plants contain the enzyme peroxidase that catalyzes the breakdown of hydrogen peroxide. Students investigated the effect of increasing the concentration of hydrogen peroxide on the activity of peroxidase extracted from radish. Okay, so he says here that the concentration of hydrogen peroxide and then the rate of reaction, how that changes the rate of reaction. Okay, so what's it called? You could see explain the effect of increasing the concentration of hydrogen peroxide on the initial rate of reaction as shown. So explain the effect of increasing the concentration of hydrogen peroxide on the initial rate of reaction as shown in figure 4.1. Okay, sure. So let's look back to the figure. You could see that as substrate concentration increases, the rate of reaction will increase up until maybe you could say 0.30, and then what's it called, after that you could see that we reach a plateau, and specifically the plateau was actually kind of reached at 0.35. But anyway, when we reach this plateau, this means that the rate of reaction is constant and that the rate is no longer increasing as you increase the substrate concentration, since the enzymes are fully saturated, or the enzyme active sites are fully saturated with substrate. And so how you explain this is you say that the substrate is no longer a limiting factor for the rate of reaction. So basically in the beginning, when the substrate concentration was increasing, that means that there's more collisions and more successful collisions between the enzyme active site and the substrate binding site, and therefore the rate of reaction will increase. Now we just write down all the information we just said. So, substrate concentration between, here you can mention 0.0 mole per decimeter cubed. Now for units, I'm just going to write x and you understand this in unit. Between 0.0 and, what's it called, 0.30, let's say 0.30, 0.30, what's it called, where's the x? Yes, and 0.30x, the substrate concentration is the limiting factor. For the rate of reaction, where increasing substrate concentration increases the rate of reaction as there are more successful collisions between enzyme active site and substrate binding site. And then you could just mention here after that after a plateau is reached, as substrate concentration, let's say that the enzyme active site is fully saturated. So enzyme active site is fully saturated with substrate, and therefore substrate is no longer a limiting factor. Concentration is no longer a limiting factor to the rate of reaction. Okay? Student determines the Km for radish, which is given to you. He says, with reference to figure 4.1, describe how they determine the Km. So the Km, or the Michaelis-Menten constant, is the value at which the enzyme reaches half its maximum concentration. Half what's it called, is the substrate concentration at which the enzyme will reach half the Vmax. Okay, so it's the enzyme value, is Km is the value of substrate concentration at which the enzyme reaches half its Vmax, or the maximum velocity. And what is the Vmax of this enzyme, we may ask? It will be exactly where the plateau is. The plateau is at 5.4, or it's at 5.6. 5.6 micromole per minute. Okay, now you're going to have to write the units on your own. I won't be writing them here, okay? 5.6 micromole per whatever, okay? So you write down your units, okay? So half of 5.6 is going to be, what's it called, using your calculator. You could calculate that. So 5.6 divided by 2 is actually 2.8. So 2.8, so 5.6 divided by 2 equals 2.8. You write your units again, and at 2.8, write your units. You can find at 2.8, if we go to the substrate concentration of 2.8, it's 0.1, okay? At 2.8, substrate concentration is 0.1, and write the units. Okay, moving on. But this is actually a very common question. Let's go ahead and describe how malaria will be transmitted by a vector, which is the female Anopheles mosquito. Now, the female Anopheles mosquito is going to basically bite someone who is infected, who is the host, who is infected with malaria, and so take out what's called the plasmodium from the host, and then the plasmodium is going to develop inside the female Anopheles mosquito, and then it's going to bite another person who is unaffected by malaria, and then therefore insert the plasmodium pathogen into his body, where it's going to obviously spread malaria. So here we're going to say the female Anopheles mosquito is the vector, which obviously here it's could, for example, bite the affected person, who take plasmodium, and which is going to develops in its guts, and then bites an unaffected person. Uninfected is a better word, actually. Uninfected person, and give or insert the plasmodium, and that's it. Okay, so this is how malaria is transmitted. Okay, diagnostic test strips for malaria contain monoclonal antibodies, and the test strips detect the antigens produced by pathogens that cause malaria. Okay, so what are monoclonal antibodies? Figure 5.1 shows stages in production of monoclonal antibodies, and the information in 3 stages is incomplete. Okay, so firstly what happens is that we have malarial antigen injected into the mouse. The mouse here is going to do an immune response, where the B lymphocytes are going to divide and form many plasma cells. So which specific B lymphocytes, which are able to produce antibodies against the specific kind of pathogen, will divide from many plasma cells, and these plasma cells are extracted from the mouse, and then there's myeloma cells, which are actually cancerous cells extracted. They're going to fuse together, so plasma cells fuse with myeloma cells, formation of hybridoma cells. Okay, now C. Figure 5.2 shows two diagnostic test strips for malaria. You have the two test strips. You have negative result, you have positive result with two strikes. Okay, a sample of blood from a person suspected of having malaria is put into a well labeled S, and a buffer solution is put into a well labeled A. The buffer solution moves the blood towards the results window, and a line at C indicates the test is working correctly, a line at T indicates a positive result for malaria. Interesting. Okay. Say 3 advantages of using test strips such as those in figure 5.2. So what I would understand here is that the antibodies are going to, the monoclonal antibodies are going to be used in this test strip. However, the question does not talk about monoclonal antibodies, it does talk about the advantages of using test strips for malaria. You have to understand that test strips in general, they're going to actually just be more accessible and more obtainable for people just to, get them, for example, going all the way to a professional and going all the way to someone who's going to diagnose you. So actually, it's not only more obtainable, it's actually also a quicker way to diagnose the disease. Another thing would be to, just stop taking all those anti-malarial drugs, which people in, for example, regions that have lots of cases of malaria, they do take these just to prevent it, just in case, without actually having to take it because they, for example, sick of malaria. So what's it called? I would say these are 3 advantages. You could also think of much more. You could talk about how, for example, these are cheaper to use. Instead of going to a professional, they could also be what else could we think about here? I guess these are enough for now because they just ask for 3. So let's go ahead and write these down. Okay, so these are more obtainable or more, accessible to people. Okay, and so make the results or some diagnosis much faster since they are more obtainable. So diagnosis is faster, and sorry about this. Okay, diagnosis is faster. And because it just is a result, it just shows directly. I think, as mentioned, one last thing would be, what's the word? Yeah, diagnosis is much faster. I think decreased use of anti-malarial drugs. Use of anti-malarial drugs. Now, here I think that you guys need to review your, spreads of malaria, the different causes of malaria, and how to prevent it. Okay, all different methods of prevention. I think this would really help you in solving questions like this because it's not the first one of its kind. There's all types of questions about advantages and disadvantages of using things like this. Okay, the highest number of cases of malaria occur in sub-Saharan Africa and Southeast Asia. Discuss the factors that determine the global distribution of malaria. Okay, so the factors that determine the global distribution of malaria, well, let's first talk about how malaria is going to spread. As we mentioned, malaria spreads by a vector, and this vector specifically lives, or the Anopheles mosquito, these live in the tropical regions of the world. So the places where there's much more humidity, the places of low altitude, the places of, so much rain, just tropical places in general. These are going to be where the mosquito lives and where the plasmodium is able to thrive and grow. So these are the areas where there's such a high distribution of malaria. And in the case where, for example, the country does not use anti-malarial drugs, does not have prevention or preventative measures, the government itself does not have preventative measures, then in that case you're going to have a more spread of malaria. Okay, so not just the country where you have a high distribution of malaria, but also other countries which do not have preventative measures against that kind of thing wherever they are. Another thing would be tourists. So for example, someone that comes who is infected with the malaria coming out into a plane where and then leaving, or for example on a plane ride where they do not really use insecticide, just in case to, kill any mosquitoes that may have come on the ride with them, because these mosquitoes could or could not contain or transfer the pathogen with them. So I think the use of insecticide, that's a preventative measure, having preventative measures of your own as a country, if you simply live in a country where you're more humid, more tropical, then obviously that will change the distribution in your country alone, and so this changes the overall global distribution of malaria. So let's start with so why it's in the sub-Saharan African and Southeast Asia is because there'd be more distribution or higher distribution of malaria in tropical regions of the world. right? More tropical regions of the world where there's more rain, near ponds, what's it called? And yeah, near, for example, near ponds, as it's where, or let's say it's just the habitat of the female Anopheles mosquito. Female Anopheles mosquito, who happens to be our vector of the day. Which is the vector for malaria. Also, that Plasmodium grows in such conditions. Plasmodium grows in warm and wet conditions. So, grows in warm. Okay, and so does the mosquito as well. Okay, so obviously when there's more of this, when there's a more tropical region, there will be a higher distribution of malaria. I think what's it called? If the country, depending of like countries having preventative measures, these will also, impact the global distribution. So if the country itself does not, for example, ban, what's it called, not like, is for example, if they decide to like not spray insecticide, if they not, they have specific clause for example, someone who comes into the country. So for example, providing these anti-malarial drugs to someone who comes into the country. So for example, some countries will provide you, as someone living in a tropical area, to first take an anti-malarial drug and to take a bunch of tests and to send out these tests being all negative and clear before you actually come into the country. So these are preventative measures. Okay, so countries and preventative measures. So when there's less preventative measures, there's a higher chance or higher distribution of malaria. Okay, so we could mention one of those preventative measures, like for example, a very famous one, which is spraying insecticide. Insecticide or pesticide in. No, I think insecticide will do. Okay, for example, in planes, and using for example anti-malarial drugs. So if these are very famous around there or not. Okay, and I think one last thing that we can mention is in here would be insecticide, anti-malarial drugs, countries' preventive measures. I think this should be it. Okay, let's go ahead and move on to question number 6. Lysosomes are cell structures that contain enzymes known as acid hydrolases. Shows the processes that occur in animal cells. So you have bacteria, you have being taken in by endocytosis. We're not really sure if it's the phagocyte or not, but there's clearly some phagocytosis going on here. Anyway, name the cell structures labeled A and E. Okay, what's A and what's E? A here is there from the plasma reticulum. As you can see, it's a bunch of membranes with some ribosomes on top. Rough endoplasmic reticulum. And then what's E? E is clearly the Golgi apparatus. And make sure to always capitalize Golgi because it's a scientist's name. Okay, state the function of the structures labeled F. Okay, what's the function of structure labeled F? Where's F? F over here is a vesicle that's moving from, using these arrows over here, from the endoplasmic reticulum to the Golgi apparatus. So I would assume it's vesicle moving, I would assume polypeptide chains from the rough endoplasmic reticulum, which we could say just RER, endoplasmic reticulum, to the Golgi, capitalized Golgi, apparatus, where I'm really sorry about all the spelling mistakes, okay, where it will be modified, okay. Name the process by which bacterial cells are taken into the cell at C. Okay, that's endocytosis, okay. Endocytosis, the folding of the plasma membrane. Okay. With the reference to processes B and D, outline the role of acid hydrolysis in lysosomes. Okay, lysosomes, let's see what we have here. Lysosomes are going to fuse with the endocytic vesicle, and they're going to release the hydrolysis enzymes, which include things like nuclease, nuclease. For example, we have, I forgot all my enzymes. Okay. protease, nuclease, and what else do we have? Breakdown of proteins, breakdown of nucleic acids, breakdown, lipase, lipids. Okay, so let's go ahead and do this. Okay, so I would say lysosome fuses with so lysosomes, I would say lysosomes fuse with endocytic vesicles, releasing hydrolase enzymes, or just releasing hydrolases, because hydrolases is, hydrolasers, fantastic. Hydrolases, which break down the pathogen or bacteria or whatever. Okay, the pathogen, break down the pathogen, just kill it, or just break down its organelles, overall kill it. Okay, which break down the pathogen, and that's could, for example, by enzymes include, you could mention examples, always mention examples, enzymes like protease, break down proteins to amino acids, lipase, break down lipids into fatty acids and glycerol. And then you can mention another example, like nuclease breaks down nucleic acids into nucleotides. Carrier proteins in the membranes of lysosomes maintain a lower pH in the surrounding cytoplasm by moving hydrogen ions. Okay, so just how carrier proteins maintain a lower pH in lysosomes, within lysosomes. Okay, carrier proteins within lysosomes, I don't think we need to get also surprised. I mean, it's just maybe that one of the lysosomes have to maintain lower pH for specific enzymes, I guess. But what I know is that to maintain this lower pH, he already mentioned the answer. He said, lower pH, it's a suggested question. So it's bound to be confusing. That's fine. But what we need to do here is, to try to write down whatever we can think about, okay? Because we're trying to gain as many marks as possible. So what I could see here is that we're talking about specifically maintaining a lower pH. How do we maintain a lower pH? He already mentioned that maintaining a lower pH would be by moving hydrogen ions. How do carrier proteins maintain a lower pH? How do carrier proteins move hydrogen ions? Active transport. There you go. Okay, active transport by how does active transport work? Moving hydrogen ions against the concentration gradient. Hydrogen ions from region of lower concentration to region of higher concentration. Lower concentration to region of higher concentration against their concentration gradient by active transport through, who comes into play here? Carrier proteins, which are going to slightly change in shape. Carrier proteins slightly change in shape with obviously a use of, by obviously hydrolyzing an ATP molecule, hydrolyzing ATP to form ADP and phosphate ion with small packets of energy, which allows it to move, it changes shape, and therefore pump hydrogen ions, and therefore maintain a low pH. Okay. Now, I would highly suggest that you guys go through this exam paper and go through the mark schemes and highlight what's important. Focus on these points, okay? Just go through these points and make flashcards of them. Okay, memorize the answers, understand how to solve scribe questions by solving as many scribe questions as possible. And so that's it. Thank you guys for watching. I hope this video was useful to you. If you have any questions, make sure to ask them in the comments section. I'll see you guys in the next one.